Welcome to the Harvest 2020 Spot Farm Results Week. The live stream session will start shortly. Before we begin, we want to remind you of some housekeeping. You will stay muted throughout the live session, so we can't hear you. The session is scheduled to be either 1 or 1.5 hours long, including questions. To ask the speakers a question, use the stage chat found on the right-hand side of your screen. Please do not ask questions for the live session in the main events chat. This session will be recorded and will be available to watch again on the AHDB Potatoes YouTube channel and the AHDB website. At the end of the session, we will provide you with a unique basis and Noroso code, which you should keep note of for each session that you attend. Don't forget to apply for points using the basis and Noroso application forms, which can be found in the reception area. Join the conversation online by following AHDB Potatoes on Twitter and use the hashtag SpotFarmResults2020. Finally, if you have any issues with the conference platform, contact the team on events@ahdb.org.uk. We hope that you enjoy the Harvest 2020 Spot Farm Results Week. Your session will now start.
Hello, and welcome to a special interactive session based around resilience through research, featuring a number of our AHDB scientists who are all specialists in their relative fields of expertise. In this open session, each of our speakers will discuss their achievements and outcomes of the work that they have done and how our overarching research plans connect with the agronomy challenges you face on farm. This is tomorrow's world, and also an exciting opportunity for you, the listeners, to use this opportunity as an open forum to feed your ideas and discuss specific challenges with our experts. This session will start with Sue Cowgill, who is our crop protection senior scientist specialising in pests. She will be covering aphids, virus and PCM. She will be followed by Alice Sin, our resource management scientist, who will be discussing the work she has already done leading on to and in relation to soils. Then we move on to Catherine Lambourne, talking about blight control research within her role as crop protection senior scientist for diseases. Joe Martin will discuss weed control and pesticide regulations, which are especially topical at the moment. And then we finish with Adrian Cunnington, Head of Storage Research, who will highlight the work that Sutton Bridge have done and an insight into the future of research opportunities. I really hope you'll enjoy this session. So now I will hand you over to our first speaker, Sue Cowgill. Over to you.
Thanks, Antonia. So what I've done with the pests work is draw out some examples of the type of research that we support, but that you might not see reported regularly at Spot Farm events. And that might be because it's work which is some sort of national monitoring platform. It might be work where we are looking more in lab-based studies about some of the key characteristics of pests. And the other reason might be that it's work that lends itself more to replicated type trials that perhaps a lot of intensive monitoring, and that they are best carried out on research institutes, trial sites. So I've picked a couple of areas, and the first one I wanted to talk about is aphid and virus. So this is an example of an area where we have a national monitoring network. So this is the yellow water trap system. It's been running for about 15 years, and it's focused towards the seed sector, providing information about the risk of virus transmission in seed crops. And that's based on counts of aphids. And we support specialists at FERA who receive samples that are submitted by agronomists and growers. The specialists at FERA are able to identify those aphids, and the information is reported through a website. So this is the map on the right-hand side. And on that map, it's an interactive map. You can go on and click on any of those regions and drill down into individual trap locations. And so you can look for a particular week, what the numbers are, but you can also look for the season so far and then compare that with previous seasons. Another type of work that we support, which is more lab-based, but it's another continuous monitoring activity, is around insecticide sensitivity testing pests. So this is an activity where AHDB, BBRO and ag chem businesses come together and support a team of researchers who, through the season, are receiving live samples from the field and testing those for any evidence of reduced sensitivity to key actives. So it's focusing on the actives that are approved at the moment, know to be effective. The peach potato aphid is their main focus, but they do other aphids. And in addition to that, in-season live testing, where we know that there's a known resistance mechanism, like pyrethroid resistance and peach potato aphid, they have molecular techniques that allow them to take dead insects, for example, from the network of traps, and test those to see what proportion of the aphids out there, peach potato aphid, is still resistant to pyrethroids. So that's another example of ongoing monitoring, and I think both the trap network and the insecticide resistance work are the type of things that are feeding into agronomist decision making in the background, but maybe we're not talking about them that frequently at spot events. Thinking about the work that we've been doing with aphid and virus just in recent history, about, I would say, 7-10 years ago, I remember there being another concern in the industry about just virus levels in crops. And at that point, there was a need for some independent evidence about what potential mineral oils offered to reduce virus transmission in a GB setting. So there are other countries where mineral oils are approved as plant protection products and being used quite routinely. For the GB, we had questions about were there any concerns about using mineral oils in relation to our seed inspection regime, because it's based on visual inspection. There's concern about whether mineral oils might mask virus symptoms. Questions about in GB conditions, are there possible risks associated with phytotoxicity? Also, does using mineral oils have an impact on ability to control blight? Does it make more favourable conditions for disease development? And then also within that work, there were trials at different locations in GB looking at efficacy of mineral oils in reducing virus transmission. So that was, I think, a starting point to generate some independent evidence for GB. The feedback we then got from levy payers was to step back a bit from it, to let the ag chems try and bring these products to market. And then with just everything that's happened in the last couple of years, with a focus back on virus again, it's an area that we've revisited. So we've looked at what information and research has been coming from other countries where they've been using it in the intervening years, whilst we haven't had work on the go. And we've used that to design trials that was carried out at the NIACOF trial site in 2020, combining some of that newer information about optimizing use of mineral oils, but also looking from mainland Europe at some of the newer ideas coming through there, things that might feel a bit quirkier, but maybe it's where we're going to have to go in the long run. And that's things like, do you need to combine some sort of physical disruption and barrier in terms of how the aphids are first finding the small potato plants, and combine that with things like mineral oils? So that was a trial that we had on go last year, and in a couple of weeks we'll have all the results in, and planning for the trial that will be carried out in 2021 in this area. Another aspect of aphid and virus that I wanted to highlight was just the feedback we've been getting about industry around questions about what independent information is there about virus symptom expression. So if I have a particular incidence of virus in my seed stock and I have a particular variety, what does that mean in terms of the symptoms, the quality issues I might see in other crops? And this was one of the topics that came up in a national forum that AHDB and NFU convened around this time last year, an opportunity for everyone to come together and just talk about experiences and prioritise issues with aphid and virus. And this area of symptom expression, one of the pieces of feedback we got was there was just a need for some updated independent evidence and information about virus symptoms and just knowledge about virus more generally. So we've updated our online content around that. So if you Google AHDB potatoes, aphid virus, there's some pages there that run through a whole range of different topics in a question and answer style. Try to answer the questions being raised at the forum. And then another area that was raised, and we're still discussing just where particular specifics of where we go in this, is this whole question of variety risk assessment. We need more information about understanding a particular virus level that we might need in terms of symptoms. So that's the aphid and virus topic. The next one I wanted to talk about is nematodes. It's obviously a topical issue at the moment. And in terms of PCN, we've been a member of the Nematocide Stewardship Programme since its inception. And that's a group that's come together to address regulators' concerns. And there's been a lot of activity from the group in terms of the training workshops that have gone in, very much led by the agri-businesses. There's been the move to have the nematicide protocol become part of the Red Tractor auditable standard. But in the background as well, there have been technical questions coming out of that group about what do we know about best practice about PCN management. And one of the areas in our earlier discussion was a whole question about soil sampling for PCN in ware crops. And that was an area where we felt there was a need for AHDB step in and fund some independent assessment of not only just what's the best approach in terms of sampling in the field, but another aspect that we looked at that perhaps we hadn't envisaged needing to think about at the beginning was just what happens to the soil sample when it gets to the lab, is whether the labs are subsampling, etc. Is that having an impact on the robustness of the estimate of PCN getting back? And it does if you've got low levels of PCN. So all that information has been drawn together in this PCN sampling and lab guide. And through that process as well, we've had a lot of engagement. There are about half a dozen PCN testing labs in GB. And we've worked with some of those, providing access to trained nematologists to address some of the questions around identifying the different PCN species, very difficult to tell apart. And we've also seen the labs wanting to come together and initiate their own efficiency testing scheme as a result of the discussions. And I think it's notable as well that some of the labs are now starting to offer different test options in terms of the amount of volume of soil that's being processed in the lab. Another area we've been working in the background on PCN is providing a PCN calculator. It's not a decision support, it's an educational tool that allows people to run different scenarios. We've got a particular starting population and different varieties that might have different tolerances or resistance or susceptibility to PCN. What does that mean in terms of final population loss? An element of that PCN work as well is decline rate. So in the absence of a potato crop, the PCN population will decline naturally. And at the moment, we don't have a particularly good handle on that. And it is known to vary, but we understand how much it varies. And one of the things that's come out of the research is quite a simple tool to measure decline rate at field level. And I'm hoping that's something we might roll out through the spot farm in the future. Just also thinking with PCN to highlight that it's not recent work, but from quite a long way out, we've been thinking that maybe do we need more information about this integrated approach, considering different options for PCN controls. So that's included work on biofumigants, but also some earlier work on trap crops. So at the time we knew about Solanum cimbrifolium, but back a few years ago, we were looking at what some of the other potential trap crops that were coming through and doing some lab screening of things like herbicide sensitivity. So that's the thinking we've been having about PCN, just that integrated approach. And then lastly, just not to forget the other nematodes. This is an area where, talking about lesion nematodes, Pratylencas, we've got a studentship that we are funding. The student Valeria is based at Harper Adams, and she's been developing some updated molecular tools to be able to distinguish the different lesion nematodes, and she's used that in combination with some soil samples submitted by growers from across England. So we've got a better idea of what the key species are. And you might say, well, why is that important? Splitting hairs. But I think for all these species, an important thing is as we move to more cover crops, just understanding maybe they potentially be hosts for lesion nematodes, some free-living nematodes. And the last thing I wanted to just draw out was this Best for Soils website, which is something that AHDB is involved in. And it's a route to be able to ask some of these questions. If you're concerned about lesion nematodes, but thinking about cover crops, this type of output just shows that there's information, for example, Phacelia is considered to be a good host for lesion nematodes. So that's the type of information that's useful in just planning some of these rotational approaches to PCN management. I hope that's been a useful run through, just to try and give you an impression that there's quite a lot going on in the background that maybe you don't see all the time through the SPOT network. It is feeding through to agronomy decision making. That's it for me. Thank you.
That's wonderful. Thank you ever so much, Sue. Just so you know that there is so much genuinely going on in terms of pest research at the moment. Just a reminder to everybody that if you have any questions, please do put them into the panel, and we'll have a full open panel session at the end of this presentation. Thank you. I'm now going to move on to Alison, who is our resource management scientist covering soils, and she will be covering the work that she has undertaken in relation to soil and water. Thank you very much, Alice. Over to you.
Thank you very much, Antonia. Yes, hello everyone. Today I'm just going to highlight some of the recent key findings from our soils and nutrient management research, and also highlight some of the practical outputs that will be coming out. Okay, right. AHDB has been funding two major soils projects lasting 5 years. These are the Rotations Partnership, which started in 2016, finishing this year, and the Soil Biology Soil Health Partnership, which started in 2017. Both are led by NIAB and involve multiple research and industry partners. The Rotations Partnership is investigating the constraints to the agronomic and economic performance of rotations, and the project is concentrating particularly on the soil physical properties that affect productivity, with a focus on potatoes in the rotation. The other partnership, the Soil Biology Soil Health Partnership, is looking to improve on-farm understanding of soil biology and to develop and validate thresholds for a range of physical, chemical and biological indicators of soil health. Both of these projects are studying trials with long-term experiments, and also investigating the effect of interventions in the rotations, such as organic matter applications and cover crops. So there's a range of measurements and assessments that are being deployed within these projects. These cover soil biology, chemical and physical indicators such as earthworm counts, nutrient analysis, pH, visual evaluation of soil structure scores, and aggregate stability. Alongside that, we're developing and testing scanning and imaging techniques such as EMI and new spectral techniques for determining organic matter quality. And furthermore, we are using yield assessments, quality and cost benefit analyses as well. The Rotations Partnership is gathering information from a number of on-farm trials, looking at the effects of different cover crops and the use of organic amendments, and results to date highlight the importance of looking at these effects over several seasons, with greater yield benefits occurring some years compared with others. With the 20 farm comparisons showed that the use of cover crops resulted in a statistically significant yield increase of potatoes about 3 tonnes a hectare. But alongside these experimental trials, as part of the rotations partnership, there has been a collection of grower survey data, which has collected information on past cropping, land use, and management practices. These encompass a wide range of soils, climatic conditions, and rotation types. Up to now, approximately 70 crop surveys have been completed, and analysis of these have shown that on average, the use of cover crops has also increased the yield of potatoes. So in both the survey and the farm comparison data, there's a numerical positive benefit of using organic amendments, but this was a bit smaller compared to cover crops and wasn't statistically significant. Economic analysis of both experimental and survey datasets showed that the increase in yield for potato crops outweighs the cost of establishing, managing, and destroying a cover crop. And all these findings will help to inform the guidance on cover crops that we'll be incorporating and will be available later this year on the AHDB website. So now I'm going to introduce the two main practical outputs from the rotations partnership. These are Trinomial UK and the Soil Health Scorecard, but these will be covered in far more detail at Soil Health webinar next Wednesday, 27th of January, which is part of a series running this year on soil health for potatoes. So please register for this webinar afterwards if you would like to find out more. So my slide is just stuck.
Yes, hello, sorry. If you could just bear with us a moment, we seem to be having a little bit of a technical issue at the moment. Just hold on a moment, please. Thanks. Alice, if you could just go on to the next slide, please. Just say you need to go on to the next slide, we'll take over.
Sorry, apologies. My slides seem to stuck.
Okay,
so apologies for that, everyone. Okay, I'll now talk about Trinomial. The UK version of the Trinomial model is being developed as part of the rotations partnership, and the model provides an estimate of the risk and severity of compaction from machinery loading. It also generates a potential stress in the soil profile, provides the contact stress in the past, and helps users to identify the most beneficial trafficking system. It can be accessed by going to Trinomial UK at the address shown on the screen. Users can input information on machinery, tyres, tyre pressure, and axle loads. The model then pulls through the corresponding soil texture data for a set of coordinates or a county, or the user can input their own. Currently, only the Scottish data is linked with the model, but England and Wales data is coming on board very soon. The tool also has the following practical uses, such as quantifying the potential benefit of using wider, lower pressure tyres, tracks or machinery with more axles. It also has the ability to display the effect of soil moisture conditions on soil vulnerability to compaction, and also allows the user to identify the weakest points in their specific management systems. So this time I'm going to see if I can go on to the next slide without causing problems. Okay, apologies, my slide seems to have stuck.
if you'd like to go on to the next slide, please. We'll take over from here, Alice. We'll just load up and deal with it. Just if you just want to just bear with us a moment. There we go. Okay, we're ready now.
Apologies, I can't seem to move the slide on.
Alice, if you just want to stop sharing your screen, we'll take over now, okay? And we'll run it from here. And then if you just basically say next slide when you're ready to, that'd be wonderful. Thank you.
Okay, I'll now talk about the soil biology and soil health scorecard, which has been developed as part of the soil biology soil health partnership. Now, there are a number of indicators which have been brought together, and these are color coded according to whether the soil assessment results where they fall within the range of expected the expected range between climatic conditions and soil types. So within the soil health scorecard, we're using a range of indicators such as visual assessment of soil structure, penetrometer resistance, and bulk density. Next click, please. And also, there are also other indicators which are readily available, such as pH, routine nutrient analysis, which is pK and mg, soil organic matter percentages using loss on ignition and total N, and also PMN, potentially mineralizable nitrogen. Next click, please. We're also introducing indicators for other biological indicators such as earthworms, mesofauna, nematodes, microbial biomass, carbon, and respiration. Next click, please. And also DNA measures of pathogens and soil health. So I just finished off talking about soil biology, soil health. The color coding allows you to decide what management decisions you need to take forward. So if it's red, the results may be just beyond the threshold in which further investigation is required. Amber means that the indicator requires further monitoring, and if it's green, then no further action is required. This soil health scorecard has been tested over 8 farmer groups, which span the range of the UK, and approximately over 100 assessments have taken place. Next slide, please. I also want to finish talking about nutrient management project, which is recently finished, looking at improving nitrogen rates through the better estimation of determinacy groups. NIAB CUF led on this work, and they have developed a practical method of assigning end groupings by counting potato leaves on the main axis. This is a quick, reliable, and rapid assessment that you can use, and is much cheaper than the previous methods of multiple nitrogen response experiments. This will enable us to provide better groupings of varieties, which will improve nitrogen recommendations. Updated nitrogen recommendations will be published in the Nutrient Management Guide RB209 with 7 new groupings to be added into the next edition, which is to be published in spring this year. there's a potential for variety plots to demonstrate how the protocol can be used where seed is limited, and also these will provide an opportunity to train users. Furthermore, they would provide information on updating and estimating groups of varieties to contribute to further editions of RB209. And that is the end of my presentation. Thanks. Sorry for the glitches.
Technology can be a wonderful thing, but it can also be a horrendous thing. So well done, and thank you very much for presenting to us today. There is one thing that we ought to talk about, and obviously with soil health being so important, Alice is actually spearheading our soils campaign for potatoes over the course of the next few weeks, and we are actually having a soils webinar next week on Wednesday the 27th of January, if anybody of you are interested. So I'm now going to move on to Catherine Lambourne, who is our crop protection senior scientist specialising in diseases, and she's going to discuss fungicide resistance avoidance strategies relating to late blight in potatoes. Over to you, Catherine.
Thanks very much, Antonia. Hopefully you can already see my slides. I wanted to talk to you today around an initiative that is a project that is being led by ADAS, but it's also got partnered with SIUC. And there's a consortium of partners in it, including many of the AgChem companies and AHDB. And the whole initiative is really around trying to gather evidence to demonstrate the application strategies which will slow down the development of fungicide insensitivities in genotypes. And so what they're doing is a range of experiments that will address the following objectives. So to determine the appropriate strategies to slow that selection using either alternation, mixtures and multi-site fungicides in multi-spray programs. They're also going to test the effect of selection of adjusting dose to the level of host resistance under field conditions, and also produce a set of evidence-based guidelines outlining best practice and cost effectiveness for the industry. So hopefully we should see some really useful practical outcomes from this work. And the work really sort of is focused very heavily around this sort of this review that was done by Van den Bosch, where you can see that there are, what he did was reviewed a number of papers around late blight control and the different strategies that were used there, and what the effect of those different strategies and changing those was on the selection of new, the development of new resistant or insensitive strains. So you can see there that increasing the dose of the different fungicides used actually did increase the selection rate for new insensitive strains. There was a slight increase as well when you increase the spray number. So we all know that the more sprays that you apply can lead to selection of insensitive strains. But things like adding a mixture partner into that, you can see had a really big impact on the decrease in the selection of that, as did the alternating or replacing different sprays in there also had a decrease on that. Whereas things like adjusting the timing, you can see, had very little effect at all on that selection process for insensitive strains. So just moving on to the sort of methodology for the project itself, there were two sites involved, one in Scotland and one in Cambridge, obviously with the two main research partners involved in that. And they used 37A2, which obviously, as we know, has already got a known fluazinam insensitivity as a tool for this. And so they used that to actually sort of monitor in the crops. They were sort of testing genotypes right the way through and used that to monitor the impacts of whether that strain was increased or stayed the same or decreased with the different types of strategies they were using in the program. So they were only comparing programs that gave good control. Clearly, we're not interested in any programs that are not going to give us control of late blight. And they wanted to test whether the following strategies would provide efficacy and resistance management. So that, as I mentioned, that sort of alternation, mixtures, and whether we were using total or half doses. And then the use of single or multi-site mode of actions, one of each. So just I've given you a list of the so I can get rid of that. Yeah, that's good. Just the treatments there. So the first one was fluazinam, so Sherlan only. Then we had mancozeb only. We used then on treatment 3 was an alternation of those with Rebus at the start, so alternating Rebus and then Sherlan. And then we went on to an alternation with Sherlan at the start and then Rebus to see whether sort of using those at different timings had an effect. Treatments 5 and 6 would use blocking, so that was three applications with Sherlan first, followed by Rebus, and then 3 applications with Rebus first, followed by Sherlan. The treatment 7 was a balanced dose mixture, so that was really looking at a sort of an opportunity then to use a half rate there, where we were using sort of both of the chemicals at half rate, and then sort of looking at them with the full rate as well, and then we've got full dose mixtures and the full dose multi-site as well. So those were the treatments that we used. I'm afraid I'm not able to go through the results of that, although we've got some intermediate results sort of from halfway through the 2020 season. The lead on this, Faye, has just asked me not to share them until we've discussed them across the group, so that they get to see them first, because clearly they're putting some funding into that. But we're already seeing some good impacts where we're different strategies that are either sort of increasing that rate of the population increase for the fluazinam in sensitive strains, so 37A2 in there, against other treatment options in there which have not had that effect on there. So we're already seeing good evidence of that. We're also doing, I think that this work will also give us a comparison of the efficacy of mancozeb containing products versus those sort of alternative or alternating strategies and different approaches we might take, which obviously with our concerns around sort of losing mancozeb in the very near future is something that we really need to be concerned about and sort of gathering evidence around that as well. The results will be available to us at the meeting. We've got a meeting due in March, and as soon as we have those in and I'm given the go-ahead, then I'm happy to say that we'll be able to share those with you as soon as we possibly can. So I think that ends my presentation.
That's great. Thank you very much indeed, Catherine. Blight is such an important disease in potatoes, and with the ever decreasing supply of active substances in our armoury against, protecting against this disease, Catherine and her team do an exceptional job. I'm now going to go and hand over to Jo Martin, who is basically our crop protection senior scientist in pesticide regulations. Very slick move on there. So we will talk to you. He will talk to you about weed research and also emus. Thank you very much, and over to you, Jo.
Thanks very much, Antonio. Hopefully you can all see my screen okay. So like Antonio says, I'm going to talk to you a little bit about some of the work that we've done over the past couple of years in terms of some weed management and potato desiccation, and then I'll move on and talk a little bit about the sort of IPM and the weed review that we conducted, and some potential areas for future research that we might want to consider for weed management. And then moving on from that, and really important and quite topical at the moment, is sort of minor use approvals and emergency authorizations. So I'll talk a little bit about that and the team that we've got here at AHDB that work on that. So if I start with the herbicide trials. So back in 2019, we did some work looking at herbicides. Two types of sites we looked at. One was looking at contact herbicides, so a replacement, and how to understand the use of replacement for diquat. So we looked at carfentrazone and how far we could push it in terms of the potato emergence. So the product we used was Shark, and currently on the label it has 10% emergence. And we obviously, when we had diquat, then we were able to go above that emergence. So it was really trying to understand the effect on the potatoes, and we did see some differences at the end in terms of yield and possibly yield effects. So it was quite clear that watching that timing of potatoes coming through was really quite critical for growers, and I think there's been some more work that's been done more recently on this as well at some of our spot farms. We also looked at that point aclonophen, so it was a new residual herbicide, and we looked at that at a number of spot sites, and we were really seeing how well it fared in combination with other effective partners. And we also wanted to look at the cost, because obviously cost has quite a big impact when you're choosing your products, and also the weed burdens as well. And what we did find with that was that the aclonophen was a really useful product, and it gave some good levels of control, and it was particularly effective as a partner product. Okay, so the next area that I want to talk to you about is desiccation. Again, this was sort of the life after diquat. So in 2019 and in 2020, work has been continuing on this. And again, it was understanding sort of the use of PPO-based products such as Spotlight or Gozai. And also we wanted to have a look at pelargonic acid and see whether or not that would be effective, as well as thinking about things like mechanical topping back in 2019, with the aim on producing some guidance on desiccants and non-chemical control in both indeterminate varieties and seed crops. And that generated some really good useful information in terms of skin set, and it also showed that both Spotlight or Gozai are effective products. Yes, there was a little bit of delay in skin set that was observed compared to diquat, but yeah, they were particular products that we could use going forward. In 2020, we really wanted to try and emphasize on hard stop situations, and there was some discussion in terms of getting the crops to naturally start to stop. So trying to reduce some of that initial nitrogen inputs onto the trials. So these were where the trials were in 2020. And we looked at reducing nitrogen levels down, and some of the mechanical methods in there as well. So I'm sure those results should be coming out relatively soon. From that sort of initial sort of look-see work within weed control and a bigger piece, we conducted a weed review across many of the crops in horticulture, in cereals and oilseeds and potatoes, just to get an understanding of where research was. And this was conducted by ADAS for us. And they came up with this sort of the IPM weed cycle. So understanding each part of a weed's development through a crop and the impact it can have, and what tools and solutions are available at each stage. And from this, there were some recommendations that came out of it for potatoes, and areas to consider going forward, including cultivations, cover crops, intercropping, non-chemical control, and also to maybe start thinking about some of the more novel and emerging technologies. So these were sort of the areas that we specifically looked at or potentially for research for the future. So it's thinking about understanding the biology of those weeds within the integrated weed management and the impact. Obviously, the question of glyphosate still comes through. We know that the Article 43 is going through renewal at the moment, so we will have to wait and see what happens with that. Herbicide resistance, obviously, that's quite key, particularly for areas where we still have blackgrass problems. And then thinking about precision application for the future and being able to maybe maintain and keep some of those products that we still have that are useful. And also thinking about mechanical weeding as well. A very specific area was volunteer potato management, and that was linking quite closely with some horticultural crops within that too. And there you go, keeping a watching brief on some of those tools for the future and seeing where they're at and trying to get that sort of that science information through from some of that commercial development that's been ongoing that will then hopefully come through for growers in different crops. So the other area I want to talk to you about is minor uses. I work with a team of 2 other people, so Joe McTighe and Adam Doxford, and we split our responsibilities by crops and also some of our expertise. So Adam has come to us from CRD. He spent about 10 years with CRD, so has good inside knowledge of the workings of what happens there. And he also looks after some of our hort crops, particularly tree fruit, soft fruit and specialty veg. Jo McTighe is responsible for our protect edibles, ornamentals, and she has particular specialism in thinking about operator exposure and risk assessments. Jo also looks after our risk register, and I'll talk about that a bit more in a minute. And then I look after most of the veg crops, but I also take responsibilities now for potatoes and cereals and oilseeds, where we see increasing demand from those sectors in terms of gaining emergency authorizations or getting understanding of some of the regulations that go on. In terms of risk register, so one of the key areas that we do is we track active substance approvals. And obviously with Brexit, then we're stepping this up even further because GB could diverge slightly from the EU approach in the future. So we look at things such as the EU pesticide database, the UK pesticide register of authorised uses. We look at EFSA journals, we look at reports that come out from SCOPAF, and we regularly liaise with most of the big ag chem companies. We have meetings with them at least twice a year to discuss products and potential issues that there may be, or potential products that might be coming through the system that would be of use. So there's quite a lot of activity that goes on in the background in terms of tracking what's been happening. And certainly with Brexit coming, or happened now, we do look at the situation as that is happening. So we're aware that approvals have got a 3-year extension, but CRD could bring that forward if another country approval comes through at a different time. So although that 3-year extension is there, those changes could come sooner. We know there's a new MRL register, which we need to look at. And we also know that there's differences now, or there potentially could be, between the EU, Northern Ireland and us in GB. So it's lots of sort of areas for us to keep track on and update people as that information comes through. The other area that I wanted to talk about that we get heavily involved in is emergency authorizations. Historically, it's something that we've done within horticulture, particularly for control of pests such as spotted wing Drosophila in soft fruit. But we've certainly been involved recently in potatoes, for example, the use of DMN for sprout suppressant, and we've currently got emergencies also ongoing at the moment, which are being considered. Now, emergency authorizations, the clue's a little bit in the title, in that they need to be reserved for true emergencies. We are aware that repeat emergencies are finding it more and more difficult to come through the system, and there is greater demand for additional data, additional stewardship, and a lot more thought and information needed to be supplied by growers to show that there has been control of that use that has been granted. That appetite to accept applications by CRD has been greatly reduced. It's something that each year we have to try and provide information in terms of what emergencies we think may be coming through before they come through as well. So there's lots of stuff that goes on behind the scenes and lots of discussions. In terms of the process, so we would work with the manufacturer and with the growers to put those applications in. They go through to HSE or CRD, who then put them forward to the expert committee on pesticides, who will make decisions. And then that goes through to the DEFRA and also the devolved governments to make final decisions. So it can take a significant amount of time, as well as effort on our part and the ministry's part to make those decisions. So like I say, we need to make sure they're true emergencies. So I hope that gives you a bit of a flavour on some of the stuff that we've been doing, and thank you very much.
Thank you, Joe. It's such an interesting but yet an incredibly important topic that you've just covered. And I'm sure many of you will have plenty of questions to put into the chat to actually ask Joe, especially in relation to the most recent casualty, which is, of course, phytate, which is a key chemical which is used in the control of PCN. I'm also then going to pass you now to Adrian Cunnington, and he is a head of crop storage research. Over to you. Thank you.
Thank you, Antonia. Good afternoon, everybody. Just making sure you can see my slides. For my session, I'd just like to say a bit about the use of maleic hydroside and its impact on storage, really. I'm not going to put up a lot of slides this afternoon, as Laura Bouvet will be talking through some of the specific results from our research tomorrow, but I do want to highlight the important role that MH has had to play in storage this season. A programme of work was initiated by AHDB on sprout suppression back as far as 2017, when we suspected the writing or maybe on the wall for CIPC. And maleic hydroside within that programme has been the subject of an AHDB review, which was published in early part of last year. And also many of you who are listening would have participated in an AHDB survey that was done as part of that review. MH was first identified in the USA as a plant growth regulator way back in the 1940s and 50s, and was first used in Great Britain in the 1980s, when it was assessed as a potential sprout suppressant at Sutton Bridge. It didn't really take off, but over the years it has gradually been adopted as a treatment for volunteer control. And in 2016, our data suggests that about 15% of the crop was being treated. Within that, 78% of you in our survey said they applied MH for sprout suppression. So overall, the treatment was given a modest score in that survey of about just over 6 out of 10 or 60% for sprout suppression, and so we were starting from quite a low base in terms of the potential for this chemical, in terms of the job we needed it to do, instead of or as well as CIPC. And at that time we started that work, we weren't really clear where that was going. But importantly, MH had some very significant potential advantages that we saw as being important for that role, particularly if we lost CIPC. Top of that list was its residual control. And what I mean by residual control is that if the chemical's taken up into the tuber, then it's available throughout the time the tuber is in store to provide control. And that is something that is not a particular facet of many of the new generation of chemicals. They tend to be more of contact type of chemicals. And so it was important to have that residual control option in any strategy going forward. Another advantage was the relative cost. At around £2 per tonne, MH was quite a low cost compared with the multiple applications of some of the new materials which were being touted at around about £4 to £5 a dose to treat a tonne of potatoes. So economics are always an important aspect of looking at any successful treatment, particularly from in commercial production. So when we suspected that future of CIPC might be in question, at Sutton Bridge we began a series of trials to look at combination treatments that included maleic hydrazide for treatment of, at that time, processing crops. So I have just one slide of that data, which shows the impact of maleic hydrazide in combination treatments. The report from this work will be available fairly shortly. The report's now being concluded, but this is data from last June's assessment of Maris Piper in this trial, which is now in its fourth and final year. You can see here the combination effect. So if we look first at the untreated bar on the left-hand side, you can see the MH3 and the MH3 in blue and the MH in green, and you can see that we get about a 50% reduction in the level of control as a result of MH treatment. But the more important bars are to the right-hand side of this graph, where we're using MH in combination with some of the newer chemicals, and in combination with ethylene, Biox-M, which is spearmint oil, and the product DMN, which is 1,4-site. You can see that the combination treatment includes
If you just want to bear a moment, please. It looks as though we have a bit of a broadband issue. If just bear with me, thank you. Right. Anyway, we appear to have lost Adrian. He has disappeared. So what I'm actually going to do now is we're going to start our live Q&A session actually, and we will bring all our hosts back if we can find Adrian. Hopefully he shall come back too, and we shall go from there. I hope everybody's actually had a really good experience of listening to our scientists and what they've been working on. I've got a few questions that have come in already, so I shall start off with one aimed for Catherine. Actually, we'll start with Catherine, and the question is: do you have any thoughts about alternative approaches to blight control using nutrition as a main driver?
It's interesting actually, because some of the discussion that we had or that the potato KEMs have been having with their spot farms has identified that they're really interested in looking at the impacts of nitrogen on their crops and what that might be doing to sort of blight impacts, etc., and incidents. Interestingly, sort of as part of the activity that we're doing within the IPM program, we've carried out some horizon scans, and that was certainly one of the areas that I came across in some research, some reported research that that nitrogen can have some effects. So it's certainly something we're going to look at, and probably through the spot farm network.
Lovely, thank you very much indeed. There is one other thing I wanted to ask you, and that came from somebody asking about, with mancozeb moving through the withdrawal process now, how important do you think the idea of spore trapping in the field will be in the future for blight control as a mechanism?
I think it becomes more and more important. I mean, we should be making, and it's something sort of as part of an IPM program that we're really keen to do, is really encouraging farmers and growers to be making sort of decisions around spraying based on data that they're collecting. I'm already involved in some similar work in the veg sector, where we do spore trapping for onion downy mildew and bremia and those sorts of things. And if you can link that with the weather data that we get through Blightwatch or other environmental tools that are available, then that really gives us clear indications of when we need to be spraying. And what it means then is that when the risk is low, we can use some of our products that are that are not multi-sites, etc., and we can save those multi-sites for when we really need them, and really sort of safeguard them for a bit longer if we can.
That's lovely, thank you. Ah, hello Adrian, you're back. Anyway, we're just starting our panel session actually, Adrian, and what we'll do is we'll actually sort of when we get some questions in for storage, we can actually sort of lead you in onto that, especially with DMN. Would you like to actually maybe just answer a question about how you went through the process to get DMN approved for use for storage?
Yeah, I'm happy to do so. Joe, you'll have to interject if you need to correct me. But yes, from the point of view of DMN, it was important for us to meet the criteria that Joe outlined about emergencies, and that was really the crux of that application that was made for DMN emergency approval. CRD were very insistent that we had control of the waste streams of the potatoes that were treated, and that obviously put a lot of control limitations on what we could do with the chemical, because clearly there's waste that leaves most potato processing establishments, and knowing where that was going and being able to make sure it didn't get into the livestock feed chain was what they were concerned about. So we had to talk with a lot of the stakeholders, the big processors, about how the controls could be guaranteed as part of the stewardship for the chemical. The other thing that we needed to do was to provide some criteria which determined that there was a true emergency situation, and not somebody just looking to try and use the chemical because it would be nice to use it. So there were criteria introduced both in terms of varieties, tonnages, and very specifically levels of maleic hydrazide residue that were specified as part of the emergency criteria. And by doing that, then we were able to narrow down the potential use sufficiently to meet CRD's requirements in terms of overall tonnage.
That's great. Thank you very much indeed. In fact, actually, I'm going to ask Joe a question because there are a couple that have come in. One was basically in terms of potential alternatives to glyphosate. And do we have any new information about what may happen to this active substance?
No, we don't at the moment. We know that the renewal is in, and we're not expecting that outcome to come out for a couple of years. So there is potential for label restrictions, I'm sure, if it does get renewed. But yeah, it's a kind of a wait and see game at the moment on that one.
Indeed. Now I'm going to move to Sue. So, Sue, talking about monitoring and managing insecticide resistance in aphids, in terms of the UK Pest project that's been going on for a while, is there a mapping element in the reporting in terms of areas and levels of insecticidal resistance in aphid populations, particularly linking to potato growing areas?
No, not in the sense of what goes on with the project is we get the samples submitted by some of the contractors on the project, and then there's an opportunity for growers and agronomists to send in samples. And so what we're looking at in the main part of the project is just, is there any evidence of loss of sensitivity to some of the key actives that we're still working with? And with that, we've not picked up any reductions in sensitivity. So there's no geographical component to that. I think one of the things we have been interested to do with some of the aphids that are relevant for potatoes, the willow carrot aphid, is we're interested in it because of it transmitting viruses early in the season, and that's one where we're actually really keen to get more samples sent in from a wider geographical base. So we are to a certain extent dependent on getting the samples in and that giving us an indication of if there are differences across regions. From the data we have so far, it doesn't look like it. In the broader project, they were also looking at cabbage stem flea beetle, not of interest here, but there you do see some regional differences. The further north you go, it's less frequent that there are pyrethroid resistant cabbage stem flea beetle in the sample.
Thank you. That was really interesting, actually. I just wanted to touch on PCN. There was a message from Keith Chappell who was asking about the PCN model. Well, I'll give you an update now. The PCN model is available, but we are actually currently developing a new one. And that will be basically running alongside the old version, and it should be up and running very, very shortly. There's also another message from John Keir, sort of mentioning about the 1 kilogram soil sampling service, and I have to say at this point that other soil testing services are of course available. I have to say that. There's a message here from Martin Cox, and he's basically asking about finding sites for Valeria and her studies. Have any of these molecular methods been utilised in labs yet?
Yes, so this is the lesion nematode work, the Pratilencus, and Martin was really heavily involved in getting the samples for Valeria. But at the moment, that hasn't been picked up. And one of the things that often comes out when I have those discussions with labs is that they're keen to get an indication of how much demand there might be for that sort of test. So that's sort of an interesting question back to the participants. People feel it's a test that they would value and want to see progress further. It'd be useful to get that feedback because it helps me with the engagement with the labs who might offer it.
Thank you. Which leads me quite nicely onto sort of looking at Alice now, and basically talking about sort of reduced tillage and cover crops, and advocating this work. Would it actually really sort of help us to encourage better soil fauna by sort of monitoring it more?
Well, this is something we've been trying to look into in the rotations partnership, but because of logistics and timings, we weren't able to follow up on FLN assessments in Rhizoctonia and Fusarium. But we're hoping to do that in the spot farms this year. Also, part of the rotations partnership, some of the growers have been involved in using the soil health scorecard. So that will involve testing the earthworms and also potentially the CO2 respiration indicator as well.
Great, that's lovely. There's a question here asking, can you please explain the Solvita test and its importance in measuring respiration levels?
Oh, I don't think I'm the best person to speak about this, because this is technically my colleague's project, but I can take that question and I'll ask her or the researcher to reply to that.
Oh, that's wonderful. Thank you very much. Right. There's a question that's come in, and I think this will probably go to Joe, this one, and that's from Asa Rowland. What kind of work is going on with potato volunteers, and what is best practice? Actually, that's a dual question between Sue and Joe, I think, actually, isn't it?
Yeah, so far, most of the work that we've been doing for potato volunteer control has been as part of our Scepter Plus program within horticulture, where we've been trying to find suitable crop protection solutions for controlling carrots and parsnips. So the team at ADAS and also at BCS have run trials for the past at least 2 years, and we have 3 potential products that we're trying to move forward with EMU applications within that area. But in terms of potatoes itself and best practice, then it's looking at the whole rotation again, isn't it? And I'm sure Sue will be able to add a bit more information in there.
So what came into my mind was just what the implication of more use of MH might be in the medium term rather than best practice. I think that might be an interesting question as to how that plays out.
Go on, Adrian, off you go.
I'm not an expert on volunteer control, but it's inevitable as a result of the need for MH to be included as a foundation treatment for storage, that the usage of MH has escalated significantly this year, and therefore we may see secondary benefits as a result of that in terms of volunteer control going forward. But that's clearly going to be, that impact's clearly going to be within the part of the production that is looking to store crops for the medium to long term, and not necessarily anybody who's either not storing or is looking for shorter term gains. So yeah, it'll be interesting to see what happens.
Super. Okay, I'm going to go to Joe now. I told you you'd be popular today. We've got a question here about, it's basically from James Lee. Hello, James. And basically he's asking, shouldn't phosphioxate be on the risk of loss list now?
That's a very good question, and I'm sorry, but I don't know the answer to that, but I'll take it away and I'll have a look.
You don't get off that lightly, because you've got another one coming now, and it begins with Z.
Oh, yeah.
Yes. So basically, do we have any current updates, having had the withdrawal of Vidate so immediately on Christmas Eve? Are there any updates on the emergency application process for this active substance
at
the moment?
Okay, so yeah, well, we were aware that we were waiting for ministerial approval back in December for the actual application, and we've been having liaisons and discussions with Corteva about it. So because there was concern, we worked with them and put in emergency applications, 4 emergency applications for Vidate, 2 for potatoes, 1 for carrots, 1 for onions. Once the decision of non-approval had come through, then CRD released those applications, and they are currently working their way through the system. The latest that I've heard back, and it's a little bit disappointing that they haven't quite made it to the ECP for January, but I'm literally reading off my phone now. Discussions are currently ongoing with DEFRA, SASA, and the respective English and Scottish ministers. So we expect to hear back from them imminently, and they'll continue to update us. So that's the latest that we've got from CRD. So we will keep pushing to get updates and as much information through as we can and as fast as we can.
Well, there you have it, listeners, hot off the press. Thank you very much, Joe, for that.
Right.
I have one for Adrian, and this has come in from Simon Faulkner. Due to the feeding restrictions on the use of DMN, it is curbing the application at the moment. Are we running the risk of not applying DMN at the optimum time this season?
We may be, yes. The optimum time for application of DMN is after harvest. So we're already in a situation where we're not in the optimum. But I think the main issue here is that some of the supply chains are having to meet a number of criteria as part of the stewardship process that I described earlier, and that is taking time. The first stewardship meeting was only just before Christmas, because that's when the emergency approval came through. There is another meeting next week, and Dawnfresh, who are the DMN approval holders, are overseeing that stewardship process. I think they are still trying to tick some boxes to meet the CRD requirements. And yes, there are some concerns that the longer that process takes, then the more we're moving away from the optimum time of applying DMN. So the usage this year, I think, will probably turn out to be quite limited, but it will still be an important opportunity to use DMN in commercial sector, which is something we've not been able to do before, and it will hopefully provide some good learning opportunities. And certainly, if anybody's using DMN, I would like to hear from you so that we can get in touch and get some feedback on how well the product's performing.
That's great. Thank you very much. Right, now we have another one for you, Adrian. You are very popular too. Martin Cox. I think one of the MH reviews picked up on the fact that MH percentage in tubers is usually the lowest in small ones. This does not bode well for control. Do you want to sort of expand on that, please?
There's certainly some work that has been done that has indicated that the MH uptake can be low in the smallest tubers. So I agree with Martin's point. Having said that, MH has been out there to control groundkeepers for quite some time, as I mentioned earlier, and I think whilst we're not necessarily going to be looking at complete control, I think it still has a very valuable part to play in that process. Again, more learning required, and until recent work was initiated, there's been very little expenditure on work on MH over the years. So some more to go and look at.
Great, thank you very much. And I'm just going to pop into Catherine now, actually, because have you got any thoughts on how the potato industry will manage in terms of blight control with the loss of mancozeb, and do you think that there are other alternative controls that will be available to them in the future?
Well, hopefully, I mean, there are a number of products available. It's just learning how best to use them and sort of using those sort of alternation strategies, etc., that we've talked about before. And I think that some of that work that I identified will actually hopefully help us see and demonstrate, you know, programs that don't include mancozeb or a multi-site in there at all, and let us sort of draw some conclusions from that as well. I mean, there are a number of different new technologies that are coming through all the time, and sort of really novel things that we sort of hope will come through, sort of things like on the genetic engineering side, where we know that the UK government now are sort of coming out of the EU. There's some opportunities for them to review where we might go with that, whether we might be able to sort of at least start some trials and sort of test out those theories. Things like RNAi, the sort of new technologies that will really impact on sort of stopping those infections happening. And on the mycete front, some of them are already you know, being used as tools in laboratories, and we see some really great effects from that, which could be completely game changing. And you know, that sounds like a really big ambition, but it is possible. It really is. That there's some great opportunities. So whilst I know that for growers, losing their multi-site is a really big challenge in the short term, but I think that it won't be long before we'll be able to look at some other strategies as well.
That's great. Thank you very much.
Antonia, could I just add in as well that we do know that UPL are expecting to submit a renewal registration for mancozeb into GB as well. So who knows if how that will go, but yeah.
Wow. Oh,
they are planning to support GB in that respect.
Wow, that's really exciting news. You can come again to another webinar. Thank you very much. That's excellent news. Right, I'm just going to finish off with Alice, and I'm just going to basically mention that Alice will actually be featuring a little bit more on soil health and doing presenting a webinar for the soil health series next Wednesday on the 27th of January. So you'll be able to hear a lot more about the work that she's doing then. On top of that, we've got Adrian will be presenting in the morning as well, so you'll be able to hear even more about what he's been doing and the important work that Sutton Bridge Storage research team have been doing. So, bringing this webinar to a close, may I say a big thank you to all our presenters, and also thank you to all for attending, and we hope you've enjoyed it and found it a great value. So if you could take the time to fill in our webinar survey, we always want to hear your thoughts, and obviously don't forget to register for your basis and Rosso points. This recording will be available shortly afterwards, and so if you have any questions, you can always contact me, and my details are on the AHDB website, and then we can actually field your questions to the relevant parties if required. Finally, you can join us again tomorrow, where we will have our KE manager, Alex Wade, hosting the morning session on reducing the threat of aphids in potato crops. And then later on, as just mentioned, we will be covering sprout suppression without CIPC with our storage team. Finally, we finish the strategic potatoes results week tomorrow with land preparation, cultivation techniques, and the importance of seed placement. This will be hosted by our own Harry Henderson. So it leaves me to say that we're very much looking forward to seeing you all again tomorrow, and thank you very much for joining us.